Assistant Professor from the Department of Biotechnology. I would take an immense pleasure to welcome you all to the third day of 1 week professional development program on avenues of biotechnology. I would like to invite my colleague, Dr. Vinod Kumar R, Assistant Professor from the Department of Biotechnology, to introduce our today's resource person.
Okay, so last stop live stream more la inga poite idu stop live stream inga vandom inda blue color idu vaanga
Sir, Kamraj sir.
Yes ma'am.
Sir, your mic is on. Kindly mute yourself, sir, please. Yes sir, thank you sir. Vinod Kumar sir. Vinod sir. Yes sir.
On behalf of our Department of Biotechnology, SRM-IST Ramapuram campus, I once again welcome all the participants and the resource person to the third day of this 1 week professional development program. And I'm very happy and immense pleasure to introduce today's resource person, Dr. K. Kasturi. She has wealth of experience in biochemistry field. She did her undergraduation BSc Biochemistry at Auxilium College at Vellore. And later she did her postgraduate MSc in Medical Biochemistry at Institute of Basic Medical Science, University of Madras, Tharamani campus. And later, after completing her MSc, she joined the same department as a PhD student in Medical Biochemistry. And after completing her PhD, she joined as an assistant professor. Since 2017, she has almost more than 5 years of teaching experience at various designations like tutor, lecturer, and assistant professor. Currently, she is working as assistant professor at the Department of Biochemistry, Tagore Medical College, Madurai. Her area of interest is very wide and it's very multidisciplinary in nature, and such as cancer biology, molecular oncology, cellular biochemistry, and pulmonary carcinogenesis. And she has numerous publications which having high impact, published in several international and national reputed journals. And she has presented number of papers or posters in national and international conferences as well as workshops conducted across the country and as well as abroad. And she also received several awards for her academic excellence. But in addition to that, she also won several prizes and awards because of our participation in extracurricular activities. And she is very actively participated in NSS activities. So I'm very happy once again to introduce to welcome Dr. K. Kasturi for our this PDP program by our department. Over to you, ma'am.
Yeah, thank you, sir. Now, without further delay, we will turn the time over to our speaker, Dr. K. Kasturi, ma'am, to deliver her lecture on regulation of GAPDH and mitochondrial dynamics treatment for NSCLC having P53 mutant gene. Over to you, ma'am.
Thank you so much. Am I audible?
Yes, ma'am. Yes, ma'am. You're audible,
yes.
ma'am.
Yes. So first and foremost, good afternoon to everyone in here. So it's my immense pleasure to be a part of this wonderful program. So my first gratitude to the institution, SRM Institute, and to the Department of Biotechnology and head of the department, Dr. Kamraj sir. So I'm very thankful, sir, for this opportunity given to me. And also very thankful to Dr. Vinod, who has given me a very good introduction about mine. Thank you so much. And my thanks to my good friend, Dr. Mani Mehle also. So without no delay, I just move into my presentation. So I hope it will be definitely very interesting and you will be definitely benefited. And like you feel free to ask any questions regarding my presentation or regarding my research. And my research part, it was like 100% in bio. But then when I move into the clinical part, like as I am working in the medical college now, I have so many clinical experiences which I can interpret into the clinical part of our medical colleges. So I have planned to give you in-depth and input regarding my research as well as to my clinical aspects. So if you have any doubt, you please stop there and ask me. I'm ready to answer you. So my research topic when I did my PhD, it was like completely about the chemotherapeutic efficacy of Nobleton. And today I'm going to give you an idea about the regulation of how it becomes like the regulation of the GAPDH in mitochondria as well as in the NSLC, which is nothing but the non-small cell lung carcinoma, which is having a mutant in the P53. Like we have so many mutations, we have so many mutant which is present in that, but especially to the P53. Why I have targeted this P53? Because it is a oncosuppressor, everybody knows. So when we were planning to do some research in cancer, we should definitely go into detail about the P53. So that is how we have concentrated on P53 mutant gene, as well as the regulation of the GAPDHs. What is the GAPDHs? How the dehydrogenase, the enzymes has been regulated, everything. Okay. So these are all introductions. Like I think everybody knows what is cancer, what are all the uncontrolled proliferation, what is mutation, what are all the mutagens. So I'm just going to give you an outline. So I don't want to give much importance to this basic parts where I have to go in detail about the clinical part. So these are all the mutagens. Whether my slides are you can view my slides.
Yes, ma'am. Yes, ma'am. We can able to view
Yes, yes, yes. Okay. So you can see here chemical, physical, biological agents. First, cancer, it is an uncontrolled proliferation of a cell. So what happened when there is any external chemical or physical or biological agents, which is nothing but a mutagen. Mutagen means it always gives a higher end of higher end of proliferation in the cells. So when it initiates, okay, so when it initiates, there is a genetical modification taking place inside the cell that leads to the chromosomal aberrations and leads to promotion. So one cell will become promoted to the adjacent and to the next. So what happened? The progression, one second. I'm sorry. Then the progression will take place. So mutation in proto-oncogene and tumor suppressor. So tumor suppressor gene will become nullified. Coming to the lung cancer, we have two types of tumors. One is benign, another one is malignant. So this benign tumor, what will happen? This will not spread. It remains in the same place where the tumor began. But the malignancy part, what will happen? It is ready to spread to the another cell, another part and another part. Then it is finally entering into the bloodstream. In final stage, it reaches to metastasis. So primarily, we have two lung cancer type. One is primary, another one is secondary. So this secondary is a very dangerous one, which is again classified into small cell and non-small cell. So here you can see, this is a classification of lung carcinoma. Like this is lung carcinoma smokers and this is non-smokers. This is adenocarcinoma and this is squamous cell carcinoma. This is small cell. Now we have concentrated on non-small cell. Non-small cell cancer, it consists of adenocarcinoma, large cell and squamous cell, all these things. Okay. So these are all the incidents where it is present and all. And these are all the stages. Stages, I think you all know what are all the primary stages, how it is forms and everything. In primary tumor, I said, like it is not affected the next organ. But in primary tumor to this next stage, the lymph node will be affected. Then it enters to the metastasis. So lymph node and metastasis will form the metastatic tumor where it is entering into the another organ, like adjacent organ. For example, liver, as well as to the brain. I'm sorry. I don't know why continuous coughs coming. I'm sorry. 1 second. So in final stage, it is ready to metastasis to the brain as well as liver, as well as to a certain extent, it is entering into the bone. So such a way, this lung cancer is highly dangerous one. So what happened when it is spreading to the adjacent organ, the distinct parts, okay, the liver, brain, heart, and bone, everything will be affected. What will happen in normal cell? We do not have the ROS, we do not have the RNS. Like you all know, ROS means reactive oxygen species and nitrogen species. So what happened? These two oxygen species and nitrogen species, they will become like radicals. They will become radicals in case of metastatic. Okay, so this free radicals will become initiated, and it is like it is entering into the growth factors. Okay, so what happened? The epithelial growth factors will be affected, so that the apoptosis level will be decreased. How it is apoptosis decreased? Because it is entering into the cell division, and it stops the cell division, so that it induces apoptosis in the treatment protocol. But what happened in the primary stage, it is reversible. So in reversible condition, all these enzymes were proliferated and all these enzymes were highly inflamed. Then what happened? It induces angiogenesis, it induces invasion, it induces metastasis. So when it is entering into the metastasis stage, by the time the apoptosis will not react and cell arrest will not become happen. So the cell division will become proliferated. Coming to the benzopyrin, it is a carcinogen. So we all know there are so many carcinogens we have, like polyamines, nitrosamines, polycyclic aromatic hydrocarbons, and nitrohydrocarbons. These are all the basic carcinogen which we can see in the human. So one such specific carcinogen is polycyclic aromatic hydrocarbon, which is nothing but a benzopyrin, which I have taken for my study. So this benzopyrin, it is present in, like you can see in the pictures, it is present in cigar, it is present in coal tar, it is present in the fried chickens and this automobile exhaust fumes, and it is highly present in the hamburgers, street chickens, and skin of the chickens everywhere. So these are the major sources for the development of this benzopyrin. Its chemical formula is C20 and H12. Coming to the mechanism of action, when this benzopyrin is entering into the body, does it actively participate? No. When it is react with the DNA only, it is activated. Okay, so we can daily expose us to all the other things. But then what happened when this benzopyrin, it is activated by the P450, like cytochrome P450, and the bioactivation of this P450 will convert it into epoxide. Then this epoxide, it is react with the interaction with the DNA. Once it reacts with the DNA, it forms dihydrodiol. So this dihydrodiol, what happens? It will form the diol epoxide, which is an active carcinogen. So what we can do in the treatment protocol, either we can stop the interaction of this epoxide along the dihydrodiol, whether to nullify the P450 bioactivation. When it is inactivated, then this will not convert it into epoxide. Or when we cut the interaction between DNA, either we can prevent it. So these are all the preventive mechanism or treatment protocol we can provide. But what happened? This mechanism, it forms the active carcinogen, leads to the cell mutation, finally leads to the carcinoma, cancer. Okay. Coming to the treatment. So the treatment protocol have chosen Noblitin as a treatment. So like we all know, so many flavonoids, so many phenols, so many anthranes, alkates. These are all the treatment organs or treatment drugs or treatment material which is used widely, my choice of drug was Nobleton. So this Nobleton, what it is doing, this is mainly concerned with the inflammation. Like we all know, cancer is nothing but some inflammatory in an abnormal form. So that abnormal inflammation can be nullified by because of this Nobleton, which is nothing but a chemical formula of C21, 22, and O8. So I think you can see there is 8 oxygen here. Okay, so this oxygen is present along with the methyl group. So this is the main advantage of this, and this is highly soluble in water. So that is how we can easily treat with anybody with the help of this. And the main source of this is, you can see here, it is from orange. So this orange is contain Nobleton of high amount, which can ready to cure the radiation therapy patient or the chemotherapeutic patient. Okay, this already we have chosen, already we have proven in vivo, but now researches is going on in the in vitro condition. Already we are doing in silico. So we are planning to continue into in vitro also. So this was successful in vivo, and in silico it is already going on. In future, we are planning for in vitro. So concerned about the Nobleton. So Nobleton, it has, as I said, like it has vital roles. So very few things I'm going to like share with you today. One is ROS. Like as I have chosen P53 as a mutant, because we have to concentrate on mitochondria. If I concentrate on all the other organelle, like this time will not enough. So I just plan of like concentrating only to mitochondria and anti-proliferation and how it is helping in the cell cycle arrest. That is all. So I told you this ROS, free radical scavenging activity, is nullified by Nobleton. Hence, it helps in the anti-proliferation. So it helps in the induction of apoptosis. So what happened? This apoptosis has to be induced by some component. So such a component is Nobleton. So we all know, when there is a deletion or mutation in the oncosuppressor gene. So for example, P53 we take. When there is a deletion, or when there is a point mutation, or when there is a frame shift mutation, any kind of mutation if taken place, what will happen? This gene will encode with the phosphoprotein. So what happened? This phosphoprotein along with the gene, it blocks the cell cycle. Okay, so it blocks the cell cycle. It helps to prevent the damage of DNA, and it helps to repair the DNA mechanism. So this DNA repair mechanism, what will happen? The division will be proliferate, and the cell is directly committed suicide by the apoptosis. This is how this Nobleton, it helps in the induction of apoptosis. So our ultimate aim is to induce the apoptosis. So once the cell induction of apoptosis taken place, obviously the proliferation will be decreased. Yes. So my target was to targeting the P53 mutated gene. So when the mutated gene, either it can be any type of mutation. So I have chosen variant type of mutations. So particularly the point mutation and the end-to-end mutation was very active in case of this diagnostic purposes. So we have seen so many cases in this, and this point mutation, they were highly active. After this P53, the apoptosis was nullified because of this. Because this Nobleton, it is helping in the cell cycle arrest. So what happened in G1 phase? So you all know G phase, G0 phase, D3 phase, and M phase and C phase. So in G1 phase, what happened? It is a highly proliferating phase where this will go and inhibit that, so that the cell cycle arrest will take place. So highly, it is involving in the tumor suppressor gene. Coming to the materials and methods. So we have chosen a 50 days old, weigh around 20 to 25 grams of mice. This is on mice. We have maintained it in the animal house with the ad libidinium, everything. So we have separated into 4 different groups, 1, 2, 3, and 4. My 1 group was control group, and 2 was only the carcinogenic group. 3 was the animal which was already bearing the benzopyrene along with this Nobleton, which is a treatment protocol. and 4 is my drug control, like to identify whether is there any side effects. So after the end of period 16th week, we have sacrificed the animal and we have collected blood, lung, liver, and kidney. Why I have collected kidney also means because kidney is the excretory product where all the end product and end metabolism will takes place in the kidney. So we have concentrated that also. Then I have coming into the histological studies and adenocarcinomas, where all the fresh tissues were experimentally assayed. So this was my RT-PCR, the primary sequence which I have sequenced it. So these are all the sequencing. I have taken P53 gene, BAX gene, PCNA gene. PCNA is for histological studies, immunohistochemistry, BCL2, caspase 3, caspase 9, MMP9, and GAPDH. So GAPDH is my like control gene, as well as I have chosen this MMP9 because MMP9, like as I told you, this P53 is along with the phosphoprotein. Okay, so this phosphoprotein, what happened? When it is entering into the extracellular matrix. So by the time this matrix metalloproteins will involve. Okay, so that is what I have chosen this. And we have statistical analysis. This everybody knows. Coming to the results, these are all basic results like how the body weight, how the lung weight, both the weights were decreased. Coming to the body weight, lung tumor, and number of animals, tumor incidence, and number of the tumor after the treatment, we have reduced the tumor incidence. Then this is the lung markers. So like we all know what are the markers we have used in like ADA, AHH, glutamate, and NT, LDH. So many markers we have used. And in clinically, if a patient comes with the carcinoma in lung, we primarily goes with the CEA, carcinogenic embryogenic antigen. So why we go for CEA rather than these ADA and all? See, aminodeaminase is also a very good enzyme. It is a marker, but then clinically they are going into CEA. Okay, so when we compare with this group 2 animals, all the group 3, group 4 animals will have a lesser values, which means it has highly influenced. Then coming to antioxidants. So I hope everybody know, like when there is a free radical scavenging, that scavenging has to be decreased because of the addition of antioxidants. That is how we always say, like I had vitamin E, which is an antioxidant. Antioxidant vitamins, the alpha tocopherol is the active form of antioxidant. That particular compound we should take. And vitamin C is again an antioxidant, ascorbic acid, and so many antioxidant things like we have external organs. So those organs will contain high oxygens. So those kind of antioxidant property will nullify the amount of free radicals. So SOD is like superoxide dismutase enzyme. This enzyme, when there is a free radicals like hydrogen peroxide, so H2O2, it is converted into H2 plus O. O is a oxygen molecule. Okay, it is a singlet oxygen where the reversal reaction is carried out by the catalase enzyme. So these two enzymes, they were vice versa. Okay, when this is increased, this is decreased. When this is decreased, that will be increased. So they both will have a combination of this. And GPX and G6PD is also like that. So glutathione peroxidase, what happened when this glutathione peroxidase? So it, like I told you, in vitamin E. So this vitamin E, when it is activated, this glutathione, yes, thione formation will happen, and this peroxidase enzyme will be activated, so that this reduced form will convert it into oxidized form. So that oxidized form will help in the conversion of antioxidant, so that the animal can be controlled with the help of these enzymes. So coming to lung and serum markers, this is also same like that. And this is non-enzymatic enzymes like vitamin E and C. Coming to the histological studies, so this is group 1, 2, 3, and 4. So you can see so many parenchymal with neoplasms. You can see, no? So this glandular pattern, okay, the epithelial cells will have a glandular pattern, and the adjoining of bronchioles, you can see the bronchioles here. So you can see the difference between these two. Okay, the non-specific chronic inflammation is higher in the group 2 animals, and after treatment, the bronchioles and epithelial cells proliferation was decreased, and you can see here a dysplasia. But here there is no such evidence of the dysplasia, and we can see some interstitial fibrosis is present in the chronic non-specific. Actually, I have compared this specimen with the human histological slides also here. So I can see the differences, like only a specific inflammation marker can be a basic different. Other than that, same the mild thickened inflammation reactions will be happened in both 2 and 4, because these 2 are controlled with the carcinogen as well as the treatment protocol animal. Again, P450, why I have targeted P450 and cytochrome B5, because I told you epoxide is converted into active form because of this P450. So we need to concentrate on that, as well as NADPH epoxide hydrolase. Why NADPH? Because we like the NAD and NADPH, which is a coenzyme form, and those coenzyme form hydrogenase enzymes will be activated. It is higher in case of cancer animals. Then coming to GST, UTP, and DTT, these are all same only. Again, cathepsin and the comparison between cathepsin D and B. Coming to the molecular genomic proteomic analysis. So just to analyze PCNA, P53 COX, MMP2, MMP9. So this matrix metalloproteins, they are from the extracellular matrix. So as well as this COX is nothing but the cyclooxygenase. So what happens? The cyclooxygenase, matrix metalloproteins, along with the collagen, everything, it is present in the extracellular matrix. So what happened in the extracellular matrix? When there is a muscle contraction or muscle dilation, what will happen? This myosin, we have both proteins, right? So we have both proteins, like protein called actin and myosin. So these 2 protein, what will happen? It is embedded into the troponin I. So what happened? This troponin I, it is a phosphoprotein. I told you it is a phosphoprotein. It helps in the myosin as well as in the actin. So this matrix metalloproteins, what happened in case of cancer condition, the proliferation will become higher. But in case of treatment animals, we can see there is a decreased amount of the proliferating agents and the proliferating epithelial cells, because the epithelial growth factor receptor, those receptor will help in the secondary messengers. Like you all know cyclic AMP, how the cyclic AMP, it is present in the matrices. Okay, so we have a receptor, we have a separate receptor for this matrix. So this matrix, it will come and bind in the receptor where it becomes activated. So what happened? This calcium cathepsin, I told you, cathepsin D and V. So this cathepsin and proteins which is present in the receptor, it will go and bind in the receptor where it becomes activated. Once it becomes activated, it intensively activated the protein kinase A. So this protein kinase A, it will again, in turn, it activates the calmodulin pathway. So calmodulin, it is nothing but an acceptor. It is an acceptor and the receptor binder, which is the gene responsible for the P53. So the gene codon, which is responsible for that, it will elevate the level of calcium, where in turn it activates so that all the enzymes will be activated. So again, the same thing, when the level of this protein, metalloproteins, when it is increases, whereby the inactivated proteins will again enter. So that will enter from the extracellular matrix and it is entering into the molecules. Then the gene and the codons will be triggered automatically, and that is entering into the post-translation modification. So you all know what is happening in the post-translation modification. So all the proteins will be entering into the translocation, where it is entering into the cytoplasm. So before it leaves the cytoplasm, the nuclear fractions will be developed. So all these things will take place inside the gene. So that is how we have concentrated on the cyclooxygenase as well as MMP2 and 9. Again, this is a PCNA immunohistochemistry, which is helping in the indicator of the malignancy, because this is, I have told you, metastatic condition. So once the patient is entering into the final stage, by the time their lesions will be like this only. So you can see this is the magnification high malignancies, and there is a immunoreactivity of the PCNA. So you can see the difference. Here there is no PCNA reactivity, but here there is a reactions with the input as well as increased PCNA proliferative cells. You can see a dot, a brown color dot, that shows the increased PCNA reactivity cells. So when compared with the second group, the third group, the level will be decreased. Okay, hyperproliferation was decreased compared to this, and in this condition, in this condition, the chemo prevention can be helped. Like we can give some like antioxidants, or we can give some sedatives, like they can have some good lifestyles, and they can have some good amount of taken up, which is killing the free radicals, everything that we can given to them. And the final thing is immunoreactivity of PCNA, like only a plain thing. You can see epithelial cells, the cells was like very good, as well as the matrices. There is no such evidence in the fourth group. Again, this is COX, cyclooxygenase. So far I have explained like how it is helping in the anti-tumor immunity, and how it is helping in the prostaglandin. Like why we have to concentrate on the prostaglandins, because PG2, where it is elevated, and then it is decreased. Okay, so that is how the cytokines and as well as the interleukins. So interleukin 6, like we all know, during the proliferation, during the inflammation, interleukin 6 will be highly elevated. Then coming to the like protein, Western blotting, as well as the gene marker for the GAPDH as well as P53. So you can see there is a difference, and you can see the graphical representation. It is nothing but like how P53 level was decreased. Okay, so we need to increase the level of P53 such a way the oncosuppressoric gene will be increased, so that it suppresses the oncogene by inducing the apoptosis, so that the cell death will be taken place properly, so that the chemo prevention will be helpful. So you can see the mRNA expressions of P53, which is a cell proliferative lung tissues. Okay, so when compare with this, we can see we can have a comparison between these two. So these base pairs, this I have given in the front slide itself, and we have compared with just a statistical Newman fuse test, and we have a graph of like how it is significant with A and B. Again, this is MMP2. Again, this is MMP9. Same only. And PCNA. PCNA is proliferating nuclear antigen. Cell nuclear antigen. Again, nuclear antigen, it is higher. Like we have seen in the immunohistochemistry only. So with this study itself is enough to prove that there is a reaction with this. But then we have chosen, like we have concentrate again into the mRNA as well as the Western blotting technique. So with that, we can come to this is Bax and BCL2. This everybody's knows how to compare the beta-actin with the Bax and BCL2, because they who are help in the main proliferation in this stage one, like how the proliferatory or inflammatory or agents which is induced the mutagens or induce the oncogene, how it is triggers. Okay, these are all the basic things. Then comes to cyclooxygenase, like in extracellular matrix, how the base pairs will be defined and how the base pairs will be based on the comparison of beta-actin. Here the beta-actin is a proliferator marker. Then this is DNA fragmentation. Like I told you, in the cleavage of chromosomal DNA, because everything is taken place in the intact in the DNA. When it is intact in the DNA, obviously the fragmented will be severe in case of bearing animals. So after that, the fragmentation will be lesser. Okay. Coming to the summary and conclusion. So I told you it has a very good alterations in vivo experimental cancers. So it maintains the clinical and physiological homeostasis, like we all know how it is maintaining, because it maintains the homeostatic animals of both the in vivo as well as in vitro. Because now we are doing actually in vitro, but then we have proved it in the in vivo, and histological examinations also confirmed about the anti-carcinogenic effect, as well as we have studied about the cellular redox status and how the confirmation and decreased productions, everything. These are all the basic things, like how glycolytic enzymes, like we all know the glycolytic enzyme and gluconeogenic enzymes, like we all know what are all the enzymes like G6PD and how it is involving and how it helps in the gluconeogenic, because the glycolysis will not be taken place 100% in case of the cancer condition. So in anaerobic condition, what will happen? This acetyl-CoA is not entering into TCA cycle because the ETC will not form. So we have 4 complexes. The complexes will become very triggering because it is highly packed, so that this will go in taking place the alternate pathway, which is a HMP pathway. So this alternate pathway they will take, and this taken alternate pathway will enter into the gluconeogenesis. Maybe the source for that will be acetyl-CoA or pyruvate or alanine, anything it can be. And it is mobilizing the retiring back molecule, and this functioning of mitochondria will be 100% based on this novelty treatment. So the membrane damage, it is basically of the malignancy cell. So we have found out like ATPase and G6, GTP, NDA, IADA, ATH, so many enzymes we have seen. Then coming to the expressions of P53, max BCL2, transcription level, in how mRNA is helping in the because after the post-transcription, the post-transcription modification will help in the removal of the uncodons. So what happened in case of cancer, like this uncodons will not be removed, it will packed again. Okay, so the uncodons will also be packed, which will lead to so many like disorders. Okay, so many autoimmune disorders also can be formed because of that. But when we given the globulin, it downregulates the mRNA expression, whereas all the codon regions alone will be packed and it is entering into the molecules. So that leads to the protein expression of cyclones. Conclusion, it will the might regulate the cell energy metabolism, proliferation, survival, invasive, etc. It suggests that globulin can highly provide to some avenues for the lung treatment. Actually, we have done it in vivo. So already, like we are doing it. Currently, we are doing in silico. We are planning for molecular docking. Already, one part of studies have completed. We are yet to describe, like we are yet to publish the paper. And in vitro, we are planning. Actually, we are planning to conduct in vitro also with the same drug. That's all. Thank you.
Thank you so much, ma'am. And participants, I request the participants can put your question in the chat box. I think some of the question it was already available in the chat box.
There is a question already available in the chat box, which is asked in the very beginning. You can go from that one.
Yes, sir. Chat box.
I can read out the first question. That is, when you were talking about the introduction, you given some foods and fruits names. So that time, one person asked, this type of fruits are taken by Western people every day, but they are not affected by cancer. Why?
This type of foods are taken by Western people, but they are not affected by cancer. Question, but they are not affected by cancer, or but they are affected by cancer?
No, I think he mean they are not usually affected by cancer. I think so. He is meaning that Western
Actually,
people
like
taking this kind of
food
foods.
They are taken. No, sir. The question is like whether they are taking the food properly, but then they are affected by cancer. Yes.
No, I mean the chart box how he mentioned is saying that this type of foods are taken by Western people every day, but they are not affected by cancer. He is asking why they are not affected by cancer, even though they are taking these kind of foods and the fruits in a daily life.
Sir, actually, like this type of food they are taken by, like whether they are daily taken, they are not affected. Yes. Why we need to tell? Like, I don't understand. See, he asked, they are taking every day. For example, orange. Western people, they are taking orange every day. They are not affected by cancer. Yes, obviously, they are taking flavonoids daily. Why is it any necessary to take affect by the cancer? No.
I think the question that he means
I think
about the
his
junk
question
food.
is not clear. Maybe, huh?
I think the question is meant to be the junk food, what we consider like a pizzas and burgers and all for Indian style of food.
I
It will
think
be
the
considered as
question is this type of foods are taken by the Western people every day, but then why they are affected by cancer? I think maybe that is the question. I think,
okay. So we'll move
if
on
at
to the
all,
next
if at all,
question.
ask Mr. Dayan, and then please type the question again. We'll move on to the next session, like next questions, and we'll definitely come back to your question.
How can we differentiate between in vivo DNA fragmentation and anomalies in practical handling during DNA isolation process?
Yes. So in vivo DNA fragmentation, like we can differentiate, because when we separate isolation, during isolation, we can understand like so many fragmentations we have. So many, like when we take an organelle, we cannot take a direct DNA. Okay. So we need to separate lysosome, we need to separate cytosolic, we need to separate every fractions, we need to separate, and then only we can understand whether this mitochondria is having, and also we have so many markers, whether this is mitochondrial marker or whether this is nuclear marker. So many markers we have. Such a way we can differentiate.
Okay. The next question, could you please highlight housekeeping genes for plants and lower organisms?
Lower organisms, I'm not sure about lower organisms, but for plants, we can take in actually this a beta HCG as a housekeeping gene, because many HCG types they have. So I think plant, we can take that as a housekeeping.
Okay. If any other questions from the participants? I think Mr. Dayanandan again put his question, like you have stated in your presentation that junk foods like burger and pizza will have cancer-related contents. So
Yes,
I think
yes, obviously. See, I didn't mention about pizza. I have mentioned about the hamburgers. Why I have mentioned hamburgers? Because the hamburger which contains the chicken, like it is steeped in the fumes, and they have a like skin of the chicken. They are highly processed meat. So those kind of meat will have a polycyclic aromatic hydrocarbon. So that polycyclic aromatic hydrocarbon, it is a carcinogen. Okay. So that is why it is it is entering into the body, and it is acting as a carcinogen, where the mutagens will formed, and it is interacting with the DNA. Okay. Next question, like the same person. Western people are taking this type of foods every day, but they are not affected by cancer. Yes. Okay. So now I got the question. They are actually taken up the foods, but then they are affected. Actually, the etiology for cancer is not only from the lifestyle or food or anything. It is like so many reasons we have. We have the genetical modification. We have so many external mutation, or we have so many what to say, UV rays. When you go for a continuous UV rays also, that will cause some mutation in the body. Okay, any type of mutation can lead to this cancer. So we cannot say that when a person is having very good diet, that person should not affect, because it can be anything. Is it your result is only for India? Actually, what I have taken, okay, this benzopyrin, it is a carcinogen. It is a polycyclic aromatic hydrocarbon. It is a carcinogen. It is for the benzopyrin. Okay, my target of benzopyrin, it is not only for the lifestyle modification, it is the genetical modification. That is what I have mentioned. It is the mitochondrial genetical. Okay, so it is not for this, it is not for India or Western people. It is for everyone. It is for everyone, but this is highly genetical modification carcinoma. That too, I have specified in the third or fourth slide, NS, like non-small cell lung carcinoma. Okay.
What about
Then
those who have never had junk or any susceptible food that can be the possible causes of their getting lung carcinoma?
what about those who never had junk or any suspect? What can be the possible causes for getting lung carcinoma? Yes, it is because of passive smoking. If a person is smoking when they are continuously exposing to the smoking, or when they are working in an industrial area, or when they are exposing into the highly polluted area. That is what I have told. Automobile fumes also it contains no. So any kind of smoke or any kind of fumes or any kind of charcoals, everything is a precursor for this polycyclic aromatic hydrocarbons. Next.
I think that's all for this question. Any other questions from the participants? If there is no question, shall we move on to the conclusion, sir? Sir, shall we proceed?
Yes, ma'am. There is any question, you can move on.
Coming to a conclusion part. And now I don't have, honestly, I have no strong enough words to express my gratitude to Dr. Kasturi ma'am for kindly accepting our invitation and deliver the talk. Definitely, this professional development program on this day would help us to expand our knowledge, started from the mutation, especially about the lung cancer, also the risk factors of the lung cancer, the nobility and the mechanism, nobility on biochemical, histopathological mechanism. Definitely will came all about today. And thank you, ma'am. Thank you for your time. And thank you for all the participants. Now the feedback
Thank you
link.
so much.
Yes. Feedback
Thank you so much.
Okay, sorry, ma'am.
Thank you, ma'am, Dr. Kasturi, ma'am. Thank you for your valuable time and efforts.
Thank you,
for,
sir. Thank you. Thank you so much.
Sharing your research with us.
Thank you, sir. Thank you so much, sir.
Now the feedback link, it will be available in the chat box. I request the participants to kindly fill the feedback link.
Ma'am, you can close the recording.
Yes, sir.
Let the meeting viable for another 5 minutes so the participants can take their feedback link. But
Yes,
you can
sir.
close
Yes,
the
sir.
recording as of now. Thanks.
Yes, sir. Yes, sir.